All right, this is part 2 of chapter 15. So we just finished talking about linked genes and Thomas Hunt Morgan and fruit flies and so on. So we take once we have identified that genes are linked, meaning they're on the same chromosome, we can take the offspring data and use it to construct a genetic map, which is basically a list of the genetic loci on a particular chromosome. And to do this, you use the recombination frequencies. And the recombination frequencies are basically the number that determines the distance the genes are apart on a chromosome. And the farther apart those genes are, the more likely they are to cross over in between. So the map distance would be further. So here's an example of creating a linkage map. You see a problem with Drosophila, and you're going to construct a linkage map. So one map unit is the equivalent to 1% recombination frequency. And you find the offspring data. And what you determine is that the distance between B and CN is 9%. And the distance between CN and VG, those are two different genes, is 9.5% there. And then the distance between B and VG, the ends, is 17%. You'll notice that this is not a perfect science. And we'll talk about in class why the difference exists between the ends and the insides. But the simple answer is because of a second crossover, a double crossover event flips the inside gene back over, and you can't see that a crossover event has occurred. But we'll detail this more specifically. Here you see a second crossover would cancel out the first and thus reduce the observed B to VG recombination frequency. So in fact, the best answer is the combination of these two individuals as opposed to this distance. So the more genes you can get in a map that you construct and determine the distance between each individual gene, the more precise it is. And so here you see some examples of actual fruit fly genes and their distance on one of the fruit fly chromosomes, brown eyes and vestigial eyes, and so on. Okay, sex-linked genes. So we've done a lot of this, so this should be pretty easy. It's determined by the absence or presence of certain chromosomes. So in humans, it's an X and a Y, of course. And the male gives an X or his Y. If he gives a Y, he determines that his offspring will be a male. And only the male can choose the sex of the offspring. Women just give an X, of course. Other organisms have different types of sex determination systems. So in grasshoppers, there's the XO system, meaning X nothing or X zero. So women are XX and males are X nothing. They don't get that second chromosome. And in fact, for men, having the Y chromosome distinguishes them. But in humans, you don't have to have a Y chromosome to be alive. You can be X nothing just like a grasshopper, but you are a female, and that's actually called Turner syndrome. In chickens, or I guess roosters or fowl, you have the ZW system. I don't know, honestly, why the difference, but you'll notice they have significantly more chromosomes. And here you have bees, where you have the haplo-diplo system, where females are diploid and males are haploid. So interesting. So there's lots of genes on the sex chromosomes that have nothing to do with sex, and these are your sex-linked traits or sex-linked genes. And they follow a specific pattern of inheritance. We always write the X's and the Y's together when doing our Punnett squares, along with the allele letters, so that we can identify distribution based on sex as well. Now, for example, some diseases that are X-linked, or sex-linked if you like, are colorblindness, muscular dystrophy, and hemophilia. Those are X-linked. Now, the other thing of note is that it is possible to have a Y-linked disorder, although that's much more rare, because the Y chromosome has only a few hundred genes on it, and the X chromosome has a few thousand. So Y-linked genes or Y-linked diseases are very rare. We've talked about this previously also. X inactivation, one of two X chromosomes in a cell is randomly inactivated during development. It has nothing to do with dominance or recessiveness. It's only in females. So here's the calico cat. Here's an allele for orange fur, and here's for black fur. It doesn't matter which one is dominant. During cell division, one of the chromosomes gets inactivated, and it's random. We don't know why one gets inactivated versus the other. And only one color gets produced, so you see this splotchy patterned look of the cat. It's a mosaic. And here's a video if you wanted to take a look at that. That explains it as well. Now, sometimes, instead of individual or small mutations that we'll talk about at a later date, like deletions, we can have large-scale chromosomal problems or issues, and that can lead to spontaneous abortions or various developmental disorders. One of the major ones is called non-disjunction, or disorders associated with non-disjunction, which means when the chromosomes split in meiosis, here, for example, is the normal splitting where this pair of chromosomes goes to the left, this pair of chromosomes goes to the right. In non-disjunction, you can have this pair of chromosomes doesn't split, so this cell gets an extra copy of a chromosome. You'll notice n plus 1, and this cell gets less. So these cells would be trisomy, or 3 copies of a chromosome, and these cells would be monosomy, or 1 copy of a chromosome, instead of the typical 2 when these eggs or sperm combine. You also notice that non-disjunction can occur both during meiosis 1, during that initial splitting of homologous pairs, or during meiosis 2, when each chromosome splits into individual chromatids. So aneuploidy is the result of non-disjunction, when fertilization of a gamete occurs in a non-disjunction type cell. And so they're going to have an abnormal number of cells. Anu means abnormal. You can think of it like that. So we mentioned trisomy. An example would be Down syndrome, or monosomy, such as Turner syndrome. Some organisms are even polyploidy, where they've evolved to have more than 1 complete set of chromosomes, and that's the only way that the adults live or survive, such as in this chinchilla. Now here's more specifically the detailed or the more precise types of mutations as opposed to large-scale changes in chromosomes. You can have minor changes to DNA. Maybe I shouldn't call them minor, but rather smaller changes to DNA, which still can have a very drastic effect. This includes deletions, where 1 or more nitrogenous bases or nucleotides is removed or deleted during replication. Duplication, when an extra nucleotide is added, 1 or more nucleotides is added. Inversion is when part of the chromosome flips around and the DNA breaks and then reattaches, so that a sequence of DNA, instead of reading AATTCC, it would get flipped and it would read CCTTAA. And then translocation is more of a large-scale chromosomal change. It's very typically seen in cancerous cells, where instead of crossing over between a homologous pair, imagine it like crossing over between non-homologous pairs. So chromosome 5 exchanges an arm with chromosome 7, which have entirely different information, and that causes major, major problems. And we, of course, don't call it crossing over. In that case, we call it translocation. Here's showing you those different things. Here's deletion, except we're not using nucleotides. We're just using the alphabet, of course, to simplify. D gets deleted. That's obvious. Here in duplication, we add an extra one, so you see BC gets copied and you have an additional BC. Here in inversion, this BCD gets flipped around and it now reads DCB backwards, which is going to affect, if that is a gene, it's going to affect what gets expressed, what protein gets made. And here you see translocation. This is a non-homologous chromatid, and A and B gets flipped with MNO. Here a breakage occurs in both, they crisscross. And now this gene carries MNO and this gene carries AB. That's called translocation. So what kinds of disorders associated with this? Down syndrome is trisomy 21. You see here pair 21. I guess I shouldn't say pair because it's a triplet. And then sex chromosome can cause certain conditions as well if you have aneuploidy, such as Klinefelter syndrome. They are XXY. They survive. They're males, but they have a few issues. Such as inability to have children, some gynecological features like enhanced breast tissue, slightly more percentage body fat, slightly lower testosterone, sometimes issues learning, and so on. And then Turner syndrome is X nothing or XO, where the male doesn't give one typically, and as a result, Turner syndrome are female, but they have slight male characteristics, or maybe let's call it less female-like characteristics. And again, they have difficulty having kids, issues with learning, and so on. Another one, cri du chat, which stands for cry of the cat. It's actually from a deletion of part of a chromosome, chromosome number 5. And they have a variety of conditions, but it's characterized by the fact that when they cry, they have this monotone, weak, cat-like cry. They also have microcephaly, which means small head, rounded face, and so on. We mentioned some cancers are associated with translocations. Here we have chromosome number 22. A portion of it gets added to chromosome 9. This has actually been identified as the the Philadelphia chromosome. It's been investigated quite closely, where part of 9 gets translocated to 22 and vice versa. So some inheritance patterns. Well, let's take let's finish on this one. We're almost done here. We'll continue. So there's some exceptions to Mendelian genetics, as opposed to the normal dominance and recessiveness that he had encountered with Ps, because not only are genes located in the nucleus, as we've previously covered, but there's genes outside the nucleus, like in the mitochondria and also in the chloroplast. So in mammals, the phenotypic effects of certain genes depend on actually which allele is inherited from the mother and which is inherited from the father. And we call this genomic imprinting. Your book will talk a little bit more in detail about that. But here we see certain genes are silenced, or they're stamped with an imprint, which involves a methyl tag that will show you through a epigenetics video on NOVA. But here you see the paternal chromosome, and this is IGF2 allele which gets expressed. And here's the maternal chromosome with the IGF2, but it's not expressed, and this is a normal wild-type mouse. This mouse is homozygous for the normal condition, and you see it's a normal-sized mouse. But then here at the bottom you see this is the mutant paternal gene, and here's the normal maternal one with an imprint, and as a result we get a dwarf mouse. So when the IGF2 gene or allele is inherited from dad, heterozygous mouse grows to normal size because it still carries the dominant allele. But when a mutant allele is inherited from the father, now the heterozygous mouse has a dwarf phenotype. So this is called stamping or genomic imprinting. It has to do with epigenetics in part. And then I mentioned extracellular genes, those found in organelles elsewhere. And in fact, this DNA is useful to trace lineage or ancestry because, for example, your mitochondrial DNA is exactly identical, assuming no mutations have occurred, to your mother's mitochondrial DNA, and always only your mother's, because the mother provides the mitochondria, dad does not. Dad only provides a nucleus from the sperm. So you can trace lineage by looking at similarities in mitochondrial DNA, and the further back you go when trying to trace lineage, the more mutations that would have occurred, and that's how you tell how similar or different you are. And there's companies that have taken advantage of this and have marketed tests. You send us a piece of hair and give us $400 and we'll send you a map that shows that, oh, look at this, your ancestors came out of Africa. What did you know? And then we talked about previously that the chloroplast and mitochondria have DNA as well. And there are genes, of course, in those that affect development and so on. And then some diseases, such as those affecting the muscular nervous system, are due to issues in mitochondrial genes. We don't entirely know too much about these, but they prevent cells from making enough ATP, and thus the reason why they affect muscular nervous systems, which require significant amounts of ATP. And we are done.